Before we get to introducing tonight's seminar speaker, what I want to do is to turn the floor over to Marina Podapova, who has a special announcement to make, and then we'll move on with our program.
Okay, so as many of you may know, there is a special scholarship established here at Iowa Lakeside Lab in memory of late Dr. John Clayton Kingston. He was an outstanding diatomist, a dear friend and colleague of all diatomists in this country, of many international colleagues. He took this diatom class here at Lakeside in 1969. His mentor was Dr. Charles Raymer. Kingston was not only an outstanding scientist, he was also a special character. As Gene Sturmer characterized him in his obituary, he said John Kingston was generous and honest almost to a fault. So he was really, basically we all loved Kingston. Unfortunately he died very young, and that's like, it was a big shock, so we all collected money, established this scholarship. So this recipient of this year's scholarship is our fantastic TA, David Birch. So please come number 9. John Kingston.
All right, and thanks for coming back, David, and doing such a good job for TAing there for the diatom course. So it's my pleasure to introduce tonight's speaker, Dr. Brent Danielson. I've been teasing Brent for the last hour and a half, trying to see whether he remembered when we actually first met. And so far he hasn't come up with it. So I'm going to give you another hint. Davis, California, ESA meetings.
Oh, 96. No, that'd be way back.
That'd be way back, like 90 or 90, maybe late 80s, anyway, a long time. So Brent comes to us via Michigan State University, where he did a BS degree in zoology. He did a PhD in systematics and ecology at University of Kansas. He then did a number of what I think of just great postdocs at some in the world of ecology, some very ecologically prestigious places, including the University of Georgia and their Institute of Ecology, including the University of Arizona. And then somehow he managed to figure out a way to get to Norway to do postdoc work, which I bet was kind of fun. I started in Iowa State University in 1992 and has been at Iowa State since then. Primarily, Brent says that he is interested in the behavior of individual organisms, particularly behavior in terms of how they respond to the environment, how they perceive their environment, which seems like it might be rather simplistic, except for the fact that for the organisms he studies, the environment is constantly changing in space, constantly changing over time, and therefore these organisms are perceiving it differently. And in response to that, they interact differently, both with other individuals of the same species that affects how their populations change, as well as interactions with other species than their own. And so he really wants to take what happens at that individual level and translate to what's going on at ecosystem level. And tonight what he's going to do is talk about mice, and he's going to talk about why they do matter in a sustainable modern agricultural system. Please welcome Brent Danielson.
Okay. Well, thanks for inviting me here. It's been a long time since I've done one of these seminars. I think the last time was probably on the order of 10 years ago, and we probably talked about gophers and why they really matter. And if you want to know what is the most important, interesting organism in Iowa, I would probably say gophers. But if you really want to know economically what was most important, I might say those guys. And we'll see why, perhaps. But this is sort of a story about how an ecologist that really would rather be out in tall grass prairies or deserts or mountainsides or somewhere else somehow gets sucked out into this kind of environment, which we all know pretty well, but maybe some of you know better than others. Do we have any farmers in the house? All farmers, I suspect, are in the field trying to plant another row of corn or something right now. But I was hoping to get a farmer or two up here because they would know infinitely more about farming than I do. I'm not a farmer. But I find it an interesting environment, at least in part because it's an environment that's totally foreign to these organisms that live out here today. And so I really want you to think about, this is an environment that might as well be Mars in some ways for the organisms that are found out there today. And knowing what they do and how they do it and how things are going and where they're progressing, I think can tell us a little bit about the future of where they might eventually go as well. So today we're going to talk about what it's like to be out in these fields and why mice really matter to sustainable modern agriculture. And I'm not going to try to produce some kind of socioeconomic model to actually show that they balance the books in a great way and make lots of profit for us. But I do want to show you that there's a potential for these organisms to be doing exactly that, even if we haven't very well quantified it. So another title for this might be 1,000 Little Hammers, stolen from a friend of mine who sort of started me down this road, Matt Liebman, who's a weed ecologist at Iowa State University. And he was interested and has been interested in the population biology of weeds. And one way you find how to make your organism interesting to people is to find out what it does to everybody else's special organism. And in Matt's case, it's weeds, and I can tell him a little bit about what mice do to weeds. So we'll talk about that. And of course, most of you know that Iowa used to be tallgrass prairie, and it's not anymore. It's a pretty desolate place relative to what it used to be. This is a particularly important little piece of it called Section 19. Those of you who are familiar with Area 51, this is the Iowa equivalent of Area 51. This is where it all happens. This is an interesting little section where we hang out a lot in the dead of winter. When all good ecologists are safely tucked in their offices, we're out there doing this. But look at that environment. Tallgrass prairie is gone. I mean, there are some remnants here and there, but they are really flecks of dust on the landscape relative to what we have out there. And even this green stuff isn't prairie. It's grassy, but it's not prairie. And so what have we done to the organisms that are there? And it's no surprise to you that if we had a really large expanse of area, we'd probably expect to see lots of species in it. And as we shrink that amount of landscape or that amount of habitat, we'd expect to see fewer and fewer species. It's sort of a regular, just a sort of intuitive thing. We don't know really what the shape of that curve might be. Well, actually we do, but it could be some kind of curve that looks like one of these. And in fact, the top curve is one that's particularly relevant because almost every organism, and here I just stole some things that I could find quickly on birds from, I think, Chile or something like that, but they all tend to show the same thing, is that if we have a really large expanse of a habitat or an environment, we have lots of species in it. And as we begin to look at smaller and smaller subsets of it, we find fewer and fewer species that were in our larger set still remaining in our smaller set. So as we take the thousands and thousands and thousands of square miles of tallgrass prairie that were in the Midwest and start to shrink them down to these little tiny postage stamps, what do we have? How many species should we expect to remain? And it doesn't really matter what species what group of species you look at. These are for birds, but if they were mammals, we'd see the same kind of curves that look something like this. They kind of look like they asymptote, but they don't actually. But they do sort of diminish in the rate of increase as we increase areas. And we can do a bunch of fancy mathematics, and if you care why people teach you mathematics, one of the reasons why is because we can find some generalities that way. And in log space then, if we were comfortable with that, we'd find that they were nice lines and that the slopes of these lines are all about the same within a given taxon, generally speaking. So that's of some value to us. But in general, as a rule of thumb, and for the purposes of just thinking about Iowa, let's think for a moment about the fact that if we had a 90% loss in the area, if we took this and we went from 2,000 square kilometers or 2,000 times 1,000, so that'd be 2 million square kilometers, and cut it down to 1/10 of that, got rid of 90% of it, we wouldn't lose all of the species, but we'd lose right around half. Okay, so as a general rule of thumb, if we got rid of 90% of some part of the environment, then we might roughly expect, based on this relationship, which we tend to see in just about every taxa we look at, about 50% of the species gone. So how much land have we lost in Iowa? And I don't think there's any doubt that we've lost 90% of the tallgrass prairie in Iowa, and probably 90% of the pothole wetlands and 90% of a lot of stuff, okay? It's not there anymore. It's now in corn and soybeans and so forth. So we should be down to only 50% of the species that we normally would have. Well, how many species have we lost, and how many species do we have to start with? And since I'm a mammologist, I'm just going to talk about mammals. And depending on how you count, we have roughly, historically, about 52 species of mammals in Iowa. You can add or subtract a few. There's a couple that we know only from owl pellets that probably flew across the Missouri River. We've got a few like armadillos that walk up here on warm summers and get hit by cars. And so they're in our collections. But generally speaking, we have 52 good species that have been known here. What ones have we lost? We lost the bison. We didn't really have as many as people might have thought to start with, but we had them. We lost the elk. We lost the wolves. We lost the bears. We lost the red-backed voles just about 10 years ago, maybe 15. One of my grad students went out looking for them in the last place they were seen and couldn't find them. And so if you really wanted to discover the remnant species, I would suggest we go looking for red-backed voles up around Pilot Mound. But those are the 5 species of mammals that, off the top of my head, I can remember that we lost, that we actually had to begin with. Only 5. So we should have lost 26, give or take, and we've lost 5. Which seems kind of peculiar to me if we've really lost that much habitat. So 4 of those weren't even lost because we took away their habitat, they're lost because we killed them. All right? Only red-backed voles weren't directly killed out. They live in forests. They're fringe species here in the very southern edge of the range. There are dime a dozen in northern Minnesota. In central and southern Minnesota, somewhat rare and patchy distributed. But they're just a relic species. So losing them probably wasn't a really big deal. It may not have even been induced by humans. It's just maybe a normal process thing. Bottom line is, somehow, somewhere, Iowa mammals are hanging on and they're doing really well. And one might suspect maybe the place to go looking is in what's 90% of our landscape, the row crop fields. And so eventually we're going to get there. But when I came to Iowa, my prevailing idea, and it's sort of sad to have to admit this, but my prevailing idea was that most of the wildlife in Iowa exists in Iowa because it lives on the fringes of the ag lands. They live on the fringes of the cornfields, the fringes of the bean fields. They live in a little bit of the old CRP ground that is now mostly buffer strips are gone for good because of a lack of programs or the rise of ethanol or a number of other things. But the bottom line was, most of us were thinking that most of the wildlife lives on the fringes of agriculture. And to the extent that they go into agriculture, we actually see them in the fields as sort of an accident or something they do for short periods of time. They go out and eat some waste grain in the wintertime like a deer does. But we don't really think of them as actually having viable, sustainable populations in those ag fields. So Matt Liebman, this guy I mentioned before, walked into my lab one day, or into my office one day, completely out of the blue and said, Brent, says, I'm studying these weeds out in cornfields. That's what I do, and we're trying to figure out what regulates the population of weeds out there. Why are there as many weeds as there are? Not more, not less. And so we put out weed seeds, and we see what happens to them, and we know that they go away. And we don't know why. But we know that if we put a screen around them, a small like window screen type of screening around them, they don't go away. Well, that wasn't too surprising, right? But if we have a larger screen with maybe 1/4 inch or 1/2 inch size holes, they don't go away too much. Some of them go away. But if we leave them out in the open entirely, then they all go away, or almost all of them go away. And he says, what in the world lives in cornfields that would be doing that? And so I thought about it for a minute, and at Michigan State as an undergrad, I'd been a student in Jack King's lab, and Jack was the man who invented Paramiscus, the genus, I think. I think he actually made them one day. He just lived and breathed Paramiscus as a study organism. And he knew that when he wanted a particular species, Paramiscus maniculatus bairdi, which is a nice flowery name to say, we call it prairie deer mouse. The place that we go to get them in central Michigan would be out in the middle of a cornfield or a bean field. So I thought about, well, there's probably some mice out there, but I always kind of thought of them, even though I had trapped them out there myself for Jack, back in the late 70s. I never really thought about the fact that there were sustainable populations out there. They were just a place where you could go out there and you catch them and you wouldn't catch too many of the other things. But what's really living out there? And so we went out to some of his study sites and we threw down some mouse traps and we started catching stuff. And we caught lots of stuff. We caught stuff that I didn't even know existed in Iowa, including this grasshopper mouse, which is a native species of Iowa, but is found in nowhere else but corn and soybean fields, as far as I can tell. I don't know of anybody who's ever caught one anywhere else. And all of them that I have caught have been there. But we caught all of these species. And all of them would be prairie species, grassland prairie species. And we caught them out in cornfields. And we caught them in bean fields. And so it turns out that we really haven't destroyed 90% of the habitat from the perspective of these mammals. Now, I won't say that they're all doing really well out there. The voles in particular, which feed mostly on leafy vegetation, don't, they look at a corn stalk, and to them it's something like a redwood tree that you would attack with a pocket knife, right? It's not going to be really profitable. And so they don't do real well where they do extremely well in tall grass prairie. But some of these other species are out there in reasonable numbers. 13-line ground squirrels, house mice are invasive species, introduced species. But some of these other guys are out there, and particularly these top two, in fairly large numbers. And so we went and we trapped a bunch of different types of fields that Matt was working on: alfalfa fields, cornfields, soybeans, and a small grain crop called triticale, which he was using as part of his study of looking at longer-term rotations. He was working with the idea that four-year rotations would be, in some ways, more beneficial to managing weeds for a number of different reasons, not all of which are obvious to me, but it comes back to some of them being related to these problems with rodents. And so if we look at the rodent abundances here, what we see is that, in fact, these long-term rotations had more rodents in them than the short-term corn, soybean, 2-year rotations that were common at the time. But nonetheless, there were still a lot of rodents even in those. And not just in the alfalfa, which is kind of where I would expect to see most of them, but also in the corn and the soybeans, where I really didn't expect to see much at all. When we look at where they are, that whole notion of, oh, they're living on the edges of the fields and they're just kind of migrating in and kind of dancing in from the edges and dancing back out, and it's really just a temporary thing, goes away really fast when we look at the dominant animal out there, the prairie deer mouse. The prairie deer mouse is mostly in the middle of the field, not in the edges of the field. They don't like the edges of the field, probably because other things like house mice and a couple of other things that are common in some woody fence rows and so forth, like white-footed mice, like a congener, Paramiscus leucopus, so white-footed mouse. They're found on those first 10 meters or so of the crops. The bottom line was, these guys are out in the middle. And by being out in the middle, that means they're occupying the most of the space. So if we were really going to talk about an organism that could have the potential for doing something out there, these guys are it. There's lots of them. They're out there in the whole field, just not just the perimeters. And they're there in abundances that, frankly, exceed what we would see if we'd been trapping the same fashion in tall grass prairie. They're not only abundant, they're more abundant than they ever were. So, an interesting kind of twist. An organism supposedly co-evolved with this fantastic grassland community, in fact, doing even better in a totally novel environment. A pretty strange thing. And so we see them out there. It doesn't matter too much whether it's corn or soybeans, and we'll see that theme as we go forward. So in fact, the real model of wildlife and agriculture is that they live in the middle of the fields, and maybe sometimes you catch them on the edges, but for at least some of the species, it's the middle of the field where the action is. So in that opening slide, at that really stark face of the moon kind of snowy field, just looking dead as can be, and you could stand there and see nothing, and you freeze to death by the way at dawn in those fields. It's colder than the dark side of the moon out there. They're out there. They're out there working. Okay, and what are they doing? So we needed to know, we needed to get some quantities on these things, and although we never got quite the quantification that we wanted, so we could sit down and write a dollar and cents, nickels and dime kind of argument for how valuable each particular mouse is, we can definitely say something about what they're actually doing out there in a fairly quantitative fashion. So that's the model of agriculture and wildlife in agriculture that I think is a little bit more prevalent than we might have guessed, at least for the small mammals. So again, we want to think about what they're doing out there. And if you went up and asked the average farmer what they're doing, oh, they're probably eating my corn. Costing me billions of dollars. Well, corn's up on top of these stalks, and it's true these mice can climb, but they're really not that capable of ripping corn off of the cob. If you ever tried it, it's kind of hard to do. They need it served up to them a little bit more, but we'll get to that. They could be consuming the crop seed that you plant in the ground, which would be a problem, digging it up and eating it. So we wanted to know something about that. Somebody else figured that out. They could be consuming weed seeds, and this was a suspicion that Matt Liebman had that brought him to my office in the first place. They could be eating insect pests. They could also be eating insects that are eating weed seeds, which would be not so good, but they could be doing that. They could be consuming waste grain, which is something I hadn't thought about, didn't think about for a fair while while working with these species. And of course, they're doing other things that from an ecological perspective are important, like becoming food for other organisms. So it's not only what you eat, it's who eats you as well. So we won't talk much about that, but keep in mind that these guys are one of the main feeders, if you will, of all the things that we find particularly interesting. And these are carnivores like foxes and coyotes and bobcats. And they're also the food for things like raptors and owls and so on. So lots of organisms that we really like have to have something to eat, and these guys are part of that. So back in the 80s, Bill Clark, a guy in my department, and coincidentally my next door neighbor, had in fact gone out there and looked a little bit about what mice were doing, because there was some concern that mice were digging up corn seed and consuming it, and each time they would pick up one of those seeds, that was a whole corn plant that didn't germinate, didn't grow, didn't produce 2 ears of corn with all that corn on it and the profit for the farmer. And so he wanted to know what they were actually eating at the period of time when the corn was coming up out of the ground, which normally would be done by now, but as we know, might extend into June or perhaps into July, the way it's going this particular year. So he had a student and went out in these fields, looked to see what was around. He saw a lot of deer mice. He saw some ground squirrels too. It's interesting, he didn't see any of the western harvest mice that I find as being the second most common species, but it was mostly deer mice, and that's mostly what we found as well. And he went to different kinds of fields that had been planted, corn on top of corn, corn on top of plowed sod, and corn on top of ground that was disked just prior to being planted. And that doesn't really matter a whole lot, but when we go and look and see what they ate, he trapped these animals, killed them, took out their stomachs, dissected the stomachs, and assayed what he found in those stomachs. What he found was lots and lots of arthropods. In fact, especially where the soil had been turned, they were consuming almost 3/4 to almost 90% of their food were arthropods. And that made sense to me because I was thinking lots of these are female mice, and female mice are about to produce litters. At least that's what our thinking was. They're about to turn on their reproduction machinery and start producing litters. They need a lot of protein, they need a lot of lipids. One really rich source of that will be the pupae of overwintering crop pests like corn rootworm and corn borer. And so that's what a lot of these larvae actually are. And that's the bulk of what they ate. And you might look and see, well, how much corn did they eat? Is a percentage of their diet? Extremely little. They weren't digging up corn seeds and consuming that. They really weren't any significant threat to the farmer's germination. When we go down and look down below, we see that they're also eating some weed seeds, including foxtail, which is one of the #1 crop weeds that we have, and velvet leaf, which is probably #2 in many areas of Iowa as a crop pest. They weren't eating much of that. They weren't eating much of that at all. And that didn't surprise me too much, but because I really thought they would be focusing on eating insects at that particular time of the year. But if we were talking about winter, these things are buried in the soil and in the leaf, in the vegetation structure. These things might be a little bit more available. So maybe they're switching their diets across times of the year. You go to the literature and see what you can find, and what you tend to see is that there are very few studies out there, because almost nobody cares about small mammals doing anything out in the fields, particularly anything good. But some people that have looked in Ohio found that if you look at just what invertebrates were doing to weed seeds over time, starting in November, when things even in Ohio are pretty cold, and in Iowa would be much colder yet, into the middle of the spring and finally the fall, the insects are eating some of those weed seeds that are out there. But if you look at how they compare to rodents, this is rodents plus invertebrates, because you can't really keep invertebrates out. If you let a mouse in, you're probably going to have beetles get in and crickets and so forth. So you can't really have insects without rodents, but you can have rodents, or excuse me, you can't have rodents without insects, but you can have insects without rodents. So what you can look at is sort of what is the difference between these curves. That's due to rodents. So what we see is that when you add rodents to the system, a lot more stuff gets consumed. Okay? And it seemed to matter a little bit whether there was residue on the field, which are the black dots, or whether the field did not have residue on it. In that case, it was the white dots. So it seemed like there may be some foraging behavior going on that might be important, and it seemed like they were doing a fair bit of work. But there's only three or four dots there for the whole year. In other words, there's not much resolution going on in that data. What Matt found, going back to his rotational plots, his 4-year rotation plots, was that when he let just the insects in, which was just this maroon set of bars, he got a little bit of predation, and it wasn't really a whole lot more than what he would get if he actually had all the seeds sort of prevented from anybody getting to them. You still lose a few seeds through the screening, but for the most part, this is just sort of normal seed loss. So the difference, the effect of insects is just the difference between these two bars. And you see insects, although lots of people like to tell you how important insects are, they're just like the most important thing. And I like to think of, well, what the hell are they doing right now in January, you know? Well, not much. But even in the summertime, they weren't doing as much as we thought. And as soon as you let the rodents have access to those same kinds of piles of weed seed, what you find is that there's huge increases in rates of mortality. So we're plotting rates of mortality here, and in some cases we'll plant survivorship in some of these. We'll plot survivorship in some of these graphs. So be a little careful of what's actually being plotted. The point is, the rodents or something large. Maybe it was birds, maybe it was rodents, maybe it was something else, but it was something large, something big enough that half-inch hardware cloth was keeping it out. They really seemed to be doing the job. So when we let them in, we saw much more mortality. And these were mortality rates, by the way, over just a couple of days. Let's multiply that by a whole year, 365 days, and it might add up to a little bit more. So here we're looking at some more explicit estimates. And again, we were still at this time worried that the edge of the field might be important. And so we put these down, I should have brought a photo of them, but they're basically some paper cards, sandpaper cards, coarse sandpaper, sort of to be like soil. We glue a known number of seeds one at a time. This is how undergraduates make hourly wages. We glue them one at a time onto these seeds, and then spray a little bit of dirt over the top of it, so that the seeds aren't really buried, but they aren't also just sticking up on the surface of the sandpaper. And it's to make them as naturally looking as possible, as though a mouse or an insect would stumble across them while walking across the surface of the soil. And you pin those down out there for a couple of days, hope that it doesn't rain, and then you go out and pick them up and count the number of seeds that are left. Okay? So that's what we were doing. And when we look at the amount of seeds removed in just a couple of days, because we didn't want to leave them out any longer because of that rain problem, we found is that when you're close to the edge, many fewer of them go away than when you're out in the middle, which tracks that distribution of deer mice very well. And as you drive around the fields today, especially the fields that aren't planted, you might notice as you get close to the edge of the field, you'll see lots more weeds on the edges of those fields right now than you'll see out in the middle. Now, there's lots of weeds out in the middle as well, but edges tend to have more. And people will say, well, that's because they're close to the source of the weed seeds that live on the edges of the field. And that might be true. But it's also true it's the place that has the least weed seed predation. So there's more weeds surviving to germinate. So we've got a little bit of animal behavior built into this. We've seen that there's some distribution of mortalities, and we're particularly interested in the fact that mortality picks up really rapidly as we get away from the edge of the field, which means that whatever is going out there in the middle is going to be the most important, because that's where most of the area is, that's where most of the crop is. And so we really want to know about that, and not just the edges. So that's what we start turning our focus to. Some of the other things we notice. Repeatedly, we see some interesting patterns in terms of how weed seeds are consumed from those cards if we put them out every other week or every week through the course of a growing season. We see that the number of weed seeds eaten jumps up really quickly in the spring sometimes, and some springs it's very slow. Why? I don't know. And I wish I knew. It's not just the females coming into reproduction or going out of reproduction and needing a different dietary requirements. There's something else going on as well, and I don't have a good answer for that. But you also notice that somewhere out here, all of a sudden, weed seed predation drops. And every year it does this, and it drops like a rock. And then the harvest happens, or at that time the harvest is happening, and we have a really hard time getting any data from late August or early September into early November. It's very hard to get data because combines and ecology aren't real compatible. They drive over traps, they drive over weed seed carts, and you never know when they're coming. Even if they tell you they're coming tomorrow, they might come today, or they might not show up for 3 weeks, and you just can't get any data. But there's something going on in this little window of time. And what we suspect was really happening was that a tremendous amount of seed is hitting the ground. Even the weed seed that isn't purposely being harvested is nonetheless being cut by the combines or raked up by the combines and then kicked out the back and settles to the ground. There's a huge influx of seed that was just this tall off the ground or maybe this high off the ground that was kind of out of reach for these guys. That suddenly hits the ground, and if you walk out into a corn or soybean field at that time of the year and just start looking around, and you got an eye for looking for weeds, it takes a little bit of time to develop an eye for it, you notice it. Man, it's just peppered. I mean, you could not get more foxtail in there than you see. Sometimes it almost covers the ground so you can't see the soil. And you say, well, I never saw that, but you weren't looking for it. And it's out there. The seed out there is phenomenal. And so something's happening at this period of time, and it's coincidental with lots of food resources hitting the ground all at one time. And one thing I should mention about these mice, maybe a couple of things about the biology of these mice. One is they don't hibernate. They do something called daily torpor. So when it's cold, during the day they don't like to go out in daylight because they die. Things kill them in daylight, so they're safer at night. But they do this daily torpor thing. So they sort of hibernate during the day, but only during the day, and then they pop back out, warm back up at night, and come out, do their thing, run around, go back home, and drop their body temperature. So imagine your body temperature, which is what, 96.8? drop to, let's say, 40. That's what they do. Okay? Now, that doesn't feel very good. Actually, it wouldn't feel like anything at all because you'd be dead at 40 degrees, no question about it. But these guys can do that. And when you find one like that, and we find them like that frequently in our mousetraps, they're sitting there kind of in a slow motion doing this kind of, like they're trying to run away from, attacking mountain lions or something. But it's all in a super slow motion because their bodies are going very slow. Well, that saves them some energy. But the bottom line is, they're going to be up and running that night just as fast as ever, running around out there in the dark. Okay, so they're active 7 days a week, 12 months of the year. Okay, they're always out there. So that's one important thing. So seed that hits the ground in October might not be eaten by them until November or January, and that might matter. And secondly, they're cachers. They're larder hoarders for the most part. Now, there's two basic types of hoarding strategies in the animal world. One is to go and put a few seeds or a few whatever you're caching, a little here, a little there, kind of stagger them all around your territory, and you can come and find them later. And the other method is you pile them all in one place. You put them in a pantry. You know exactly where the pantry is. You put your back to the pantry, defend it from all comers, and that's your grocery store for the rest of the winter. They do a little bit of both of those things. Okay? And we'll get to see a little bit about what that looks like. But the important part about caching organisms is they tend, as a rule, to cache far more than they can use. Because they really don't know how much they're going to need. So imagine somebody told you that there will be no more chocolate for we don't know how long. Okay, are you going to put one package of chocolate in your closet? No, you're going to buy boxes and boxes of it, right? And put it in your closet. And then you might put in 10 years worth of chocolate in your closet because you don't know when chocolate's coming back. So, okay, so that's what these guys are doing, which means that they're basically insatiable carnivores. Even though they have tiny little stomachs, they eat far more than we would think because they don't actually have to eat it. They just have to take it and put it someplace where it can't grow. So those are important things to remember. These guys are active all year round. They're collecting seeds all year round. They're caching seeds all year round, but they're also living off their caches when they can't collect something if, for whatever reason, they can't find something or whatever. So bottom line is they're really voracious little eaters. And yet something's happening late in the season and we didn't know what. So I want to show you one more thing that happens, and that is, you might say, well, what happens when a lot of seed is on the ground or in weedy, seedy places? We could have a lot of mice in there, and it could be, if you were a seed, the last thing you want to be is in a patch with a whole bunch of other seeds, because every granivore out there is going to come looking for that patch, and when they find it, they're going to eat everything in it, and you're gone. You're done. You're whatever you want to call it, whatever the seeds say when the time their number has been called. That's what's going to happen to them. On the other hand, it might be really good to be a seed way out in the middle of nowhere, all by yourself, because nobody would come looking for you out there. And somebody might stumble across you by accident, but you might kind of be safe out there. So it's possible that the distribution of seeds might be really important and the abundances of them. On the other hand, if there's a whole lot of seeds around, then maybe everybody else gets eaten and you don't. And this is a sort of African ungulate herd hypothesis, if you will. We see all of these zebras and wildebeest standing around together. Why do they stand around together? They stand around together because, well, if there's a lion out there, a lion's going to eat somebody. And if there's 100 other things out there, there's only one chance in 100 he's going to eat me. Okay? And so I want to stand around a bunch of things. So maybe that works. Maybe you do, as a seed, want to be in a big pile of seeds because then you're protected. So we needed to answer that question by going out and putting lots of seed in small patches out there, small patches being about the size of this room, and seeing whether or not we got increased weed seed predation or not. And the answer was, looking at those things, well, hard to tell. And I could go through all of those different curves and tell you about each one of them, but I'd rather not, because there's really not a lot of pattern there, except that we see this kind of, if there are really lots of predation in the middle of the summer, it drops off at the harvest, or maybe it just stays low the whole time. Why weren't there lots of seeds sprayed upon in these fields? We don't really know. Bottom line is we got lots of different responses. On average, it appears that if you put out lots of seed, you'll probably get more seed predators, get more mice, and you'll get more predation. But it really wasn't very clear. So we needed to investigate that a little bit further. And so we'll take a look at that. But I want to go back and talk a little bit about what the mice were doing. And the mice, of course, normally live in tall grass prairie like this. And if you think about a mouse down there on the bottom of vegetation that's this high, and you think about the fact that a red-tailed hawk or an owl would like to feed upon them, catch them, or a coyote or a fox would like to find them, they're pretty hard to find in here. Even if they're above ground during the day, they're hard to find, never mind at night. Okay? So from a mouse's perspective, they're pretty safe from predators, we might think. And they're at least some of the predators. Snakes, not so much. Weasels and shrews, not so much. But from the bigger predators, they're pretty safe. There's, of course, different times of the year when they're not so safe. We get occasional fires, and they're running around on this rather dark-colored ground. It has a lot of charred material there. So that might be the only time they're really exposed to predators from the air or from above anyway. But when we look at where they're living in cornfields and soybean fields, this is what they're contending with. Okay? Lots of open time, lots of open space, very little protection. And you might notice that these fields are kind of different in color. There's black parts of the field and there's really light colored parts of the field. And that might make a difference because they're more vulnerable on some certain colors against certain backgrounds than they are against others. So particularly that dark background, the mice themselves are fairly dark and they blend in pretty well. But on that hillside, maybe not so much. So we might learn a little bit more about the micro-distribution of weeds by looking at the foraging of these things. And lo and behold, along comes a kid that wanted to go to grad school and wanted to study mice in cornfields. And I couldn't say no to this. So I took on John Dowden as a PhD student, and he really likes animal behavior. He likes doing experiments on individual animals. He stole a couple bird aviaries from an ornithologist, filled them up with dirt, put little mouse-proof barriers around them, and then proceeded to manipulate the backgrounds and find out, where do these mice actually feed? Do they prefer to feed in dark soils? Does it really matter? Because if it does, then seeds on dark soils are going to be more vulnerable than seeds on light soils. Or maybe it doesn't make any difference. And he did a lot of different experiments, so I'm just going to show you one of them. He did this with different light intensities. He was able to string up basically little LED Christmas lights, put a rheostat on them, adjust the lighting so we had something that looked a lot like a clear sky with no moon in it, and a clear sky with a full moon in it, and all the things in between if we wish to. And you could put different types of seeds, velvet leaf and foxtail seeds, and these give you something of a rough idea of what the differences in size here are. You can see these things are heart-shaped and they're quite a bit larger than the foxtail, which are about pinhead size. So these are about 3 or 4 times the size of a pinhead. Put those seeds out there and see whether it mattered where the seed was, what kind of seed it was, what the light environment was, and just learn a little bit about the foraging ecology of the mice. And he created these little runways, if you will, some on light-colored soil, some on dark-colored soil, and he put seeds in these trays and he measures how many seeds are consumed. We call this giving up density, the number of seeds that are left. And that tells us a little bit about the motivation of the mice. If the mice are really motivated to feed, they'll depress the levels of seed that are left when they're done foraging to very low levels. If they're not motivated to feed a lot, perhaps because they feel like it's a dangerous environment, then they'll leave more seed behind. You can think about this as, if you knew there was a man-killing, Siberian tiger roaming around Lakeside lab property, and you were going to go out and look for morel mushrooms, a month ago when they were out, you probably wouldn't look too hard and you probably wouldn't gather too many morel mushrooms before you decide, I think maybe it's time to get back to the cabin, right? But on the other hand, if you felt pretty safe out there, you might stay out for quite a while and consequently catch quite a few mushrooms. Well, what we found was that for both of these two species of weed seeds, indeed something like that was happening. Lots of seeds were left when we were looking at a light-colored soil. Those are the light-colored bars. Because the mouse felt basically threatened. It could be killed, it could be picked off there more easily. And it doesn't want to expose itself to this. Now this is an organism that normally lives underneath a grass canopy, which it's just not really going to be normally worried about that kind of thing, but somehow it has the ability to understand that there are times when we need to worry about this, and it knows how to adjust its foraging accordingly. What we also see is that there's more foraging going on when the lights are off than when they're on, but in fact it didn't turn out to be particularly significant. It was on the edge of significance, if you will, of going that direction. Lots of other studies on other organisms of other small mammals show that light levels really matter, but in our case we didn't come up with a statistical sense of that, although it was trending that direction. We also see that in general foxtail is foraged on much more than velvet leaves. Velvet leaf are large. You'd think they'd be tastier, but it turns out they're really hard. They have a very hard seed coat on them, and so they're hard to open up. And they also are probably not as tasty. I think they have a fair number of toxic compounds, but I have no way to actually extract them. So I'm speculating a little bit on that. Whereas I think the foxtail is pretty much 100% digestively good stuff, if you will. So they eat quite a lot of it. So we find that in these kinds of environments, they forage differently, which might tell us something about the patchiness of weed seeds. Might also be a good thing that they prefer to forage in these dark soils, because if you look out there, a lot of times where I see the most weed seeds are the low spots that tend to be the darker soils. So maybe the mice are going to focus in those areas. Hopefully that would be something that would aid in regulating those weed populations to some extent. We also started getting concerned about, well, what do they really prefer? That last study showed that they really preferred foxtail to velvet leaf, but what about some of the other things out there? In particular, what about the grains that we know that are out there, either the soybeans or the corn? And so one of the things we did is we brought mice into the lab for short periods of time, and we introduced them to very simple dishes full of stuff. They have their choice. They can eat, in this case, velvet leaf or corn, and we measure how much they feed on those things. And we can do all of these species in a pairwise fashion, and we did that, and I apologize a little bit for this slide. It's a little hard to read, but when we do that, just like we saw in the previous experiment, foxtail, when offered side by side with velvet leaf, is a far preferred seed. So we might suspect that there's more potential for mice to regulate foxtails, at least from the biology of the mouse, than there is velvet leaf. Now, that's not entirely true in terms of the regulation part, because we really don't know how much mortality these two species can withstand. So this much mortality might be much more important to velvet leaf than that larger amount is to foxtail. But the bottom line is they really like foxtail. When we compare foxtail to corn, however, look at that. It's definitely not a preferred food relative to corn. And in fact, velvet leaf isn't either. And then when we compare foxtails to soybeans, we also found that soybeans are highly preferred to either of our weed seeds. And this suddenly began to give me a little bit of a thought about that plunge in weed seed predation that happens at the harvest time. At the harvest time, when most of the weed seeds hit the ground, all that waste grain is hitting the ground too. And there's tremendous amounts of waste grain in these fields. An average of something like, I don't know, 350 pounds per acre or something like that. I can't remember the numbers off the top of my head, but I went to the USDA to get those numbers. And the bottom line is that they're eating lots and lots of this stuff. So we developed this idea that maybe when we see the harvest, we're seeing this moment when food is suddenly hyper-abundant. And these guys can go along and skim the cream, the best stuff off the top of that first. And so they probably are focusing on waste grain and not the weed seeds that Matt Liebman had been studying in September and October and November. They're not taking those because there's this other stuff out there that's so much better. Wouldn't you take that first? If I put a piece of chocolate out and I put, I don't know, a soy burger out or something, which would you take? Well, you'd probably take the chocolate first, right? And so that's what they're doing, okay? In midwinter, maybe they switched to feeding on weed seeds because they've got all the waste grain out. Now, I believed that at one time, and now I'm not quite so sure that's true, but certainly they might switch to taking at least some more weed seed later in the winter, depending on how much waste grain is around and still available to them. So there might be a switch that goes on late in the winter. But if you're a weed seed, you hit the ground in late September, early October. You've gone through a bunch of freezing and thawing cycles, probably some rain, a little bit of snow. Maybe there are some crop management things going on that also might stir you into the soil. But the bottom line is you've had a few months to escape, to run from wherever you landed on the ground to somewhere down in the soil where you might be safe. Now the mice will dig in the soil to get weed seeds out, but they don't dig very far, and they're not going to find you as easily if you're buried in the soil, even if it's just a little bit. So if they did switch in January or February, that might be too late to catch some of those weed seeds that they would have otherwise caught had they been hunting for them earlier. So that became our sort of working hypothesis. We went out to the field to find out, well, how many mice are out in there? And does it matter what kind of field we look at, whether it's a cornfield or a soybean field, is there differences? And suppose that they don't like soybean fields at all, which is kind of what I thought, because those are so desolate, there's just no crop residue, there's just nothing out there. How in the world can they survive out there? Can't be any mice out there. And I thought, well, if there were no mice allowed during soybean years, then you only get 1 year of corn, then it goes back to soybeans, or at least that was the rotation up until recently. So maybe you'd never get to build up very big mouse populations in cornfields. Yes, they're out there, but maybe not lots of them. So we had to go out and start trapping them. And what we found was that in general, there's a somewhat fewer, maybe 2/3 as many mice in soybeans as there are in cornfields, but there's not a huge difference. So we don't have, wasteland in the terms from the perspective of the mice. They're out there, they're doing okay. This is fine stuff. They prefer cornfields a little bit more, at least in terms of abundances. And we also found that it didn't matter whether cornfields and bean fields were tilled in the fall or not. And this surprised me. I thought that the tillage would really make a difference. I thought they would at least need some of that corn detritus to hide under, to stay protected from predators, and also to stay a little bit protected from a very cold winter sky, which is sucking heat out of their tiny little bodies, which have really large surface areas for their body size. They cool down really fast. It takes a lot of energy to keep them warm. So I thought that plant cover would be really important, but as you can see, tilled or untilled didn't matter at all. And it's kind of interesting to live in a land where literally your whole neighborhood goes upside down, and it doesn't matter. These guys don't care. You plow them, and they're right there the next time. Not a problem. So they're out there in abundance. The next thing we did was we started looking at, well, what happens to weed seeds over the winter. We've been looking at them in the summer. We've been looking them up into the harvest time. But what if we actually went out in the field after the harvest, which is kind of hard to do because farmers, university farms, aren't very good at telling us when they're going to be done with the field. But when we finally get out there, we throw down a bunch of seeds and we leave them there, walk away, come back in the spring and dig them back up again. How many do we recover? Well, it's a little bit tricky because how do you know that you actually found them after laying out there all winter? Maybe some of them blew away. Maybe some of them floated away in a good hard rain. So it took a little bit of technology to do that. And one of the means of doing that was to come up with some beads that were the same size and density as the weed seeds that we were interested in. And we put those out there. These are plastic beads. And the importance of that is that if in fact the weed seeds were blowing away or floating away, these beads would act just like those weed seeds, and they would go away too. And so the difference between the weed seeds and the beads would be the amount that was taken by something else, presumably the predator, because predators are smart enough not to eat plastic. These mice aren't that dumb, so they don't eat plastic beads. So what we found was that, okay, mean numbers of beads retrieved is pretty small, or is rather pretty large, so they're not going away too badly. They're not too much different in our different 2-year corn rotations and soybean rotations, or our 4-year rotations in those different crops, all about the same. But when we compare and then correct for the number of velvet leaf seeds that are actually lost by predators, we find tremendous amounts are lost whenever they're exposed to things that allow, to the situation which allows mice to come in. So in these yellow bars, they're basically open to the sky. Okay, so mice can come in. These smaller ones have screening over them, either that excludes everything or excludes small things. Sham exposures, which basically act like the larger exposure, except they have holes in them. So the bottom line is that there's large predators that are somehow taking these things out during the winter. Weed seeds are being lost over winter. When it's snowy, when it's cold, when it looks like nothing's out there, in fact, something's taking these weed seeds away. So, in fact, what happens after the harvest is important. Whatever's happening, the weed seed mortality is ramping up as we go further into the winter. So along comes another grad student who likes to work in the winter time. This is Tatiana Flick, a master's student. She also was totally motivated, totally driven to study agroecology. She's from Canada. What better student could you have for working in the middle of a cornfield than a Canadian? This gal is bulletproof. She can go out in the coldest weather. And she's just one of the best field ecologists I've ever seen. She loves working with mice out in these kinds of environments. And she wants to go out there and figure out, well, what's going on in those fields? How is the distribution of the seeds out there really matter? How does it really matter? And she's particularly interested in waste grain. And one of the interesting things about waste grain is we've created this corn, which has this built-in herbicide resistance. And we can't get it out once we got it in, because we can't go back with Roundup and spray the field and kill this stuff off, which is now a weed, because we don't want it there. And it turns out they're a weed even in corn crops. If we were to plant corn on top of corn, this feral corn will in fact reduce your yield by about 13% under some conditions. So it's even bad if you were going to plant corn into corn. These plants get in the way, they reduce your yield overall. And those guys love nothing better than corn. So she puts out corn in different kinds of arrangements, different kinds of patches, and she's looking to see, well, how much volunteer corn comes back. And we can't really measure sprouting yet, we will, but we can measure how much corn is left over at the end of the winter. And when we do that, what we find is that these guys are pretty good at it. We know they do that. They respond to the patchiness in some species of mice in Europe, but they also do it with the Paramiscus here in Iowa. And so I'm going to move right down here to her experimental design. She basically made lots of small patches. So these yellow areas are patches of high-density corn, okay, where she's added corn to a field which has no corn in it, and larger patches. The experimental plots are the same size, but the arrangements of the patches are a little bit different. And she's hypothesizing that we could see, in some cases, lots more mice move in. Now, maybe we could get more individual mice in a situation like this, and we can't hear where they'd be crowded together. Okay, so numerically, we might see a different response in the mouse population. But we might also see that they forage more efficiently in some situations. So individual mice will do better here maybe than here, and then we have our control to compare to. She puts out nest boxes, and the important thing about nest boxes here, there's two of them put out side by side. One of them becomes their apartment. They put their bed in there, they have a kitchen over there, but this is the pantry. Look what they do, they fill this. This happens over and over again. You put two of these nest boxes side by side. They fill one of them completely full of corn, and the next one they use as a living space. So now we can measure how much corn is actually being cached. They fill every box you put out there. But what we find is that, in fact, the treatments don't really matter in terms of the number of mice that are out there, whether it's the number of first captures, which would be an estimate of the number of individuals, or the activity of the mice as a whole. In these three different treatments, we seem to have about the same number of mice running around every night, doing about the same number of things. One of the things we did notice, though, is that we get a lot of reproduction. And that's something we hadn't expected. So that's just a little aside. We're not going to go anywhere with that. But these guys are reproducing in cornfields in the dead of winter. Mice don't breed in winter, except in cornfields. So they're really doing well in those cornfields, much better than we had anticipated. We get a little bit of a difference in terms of their foraging behavior. It's not particularly strong between the different treatments, but it's a little bit stronger in both treatments than it is in the control. But when we look at how much grain is actually left in the field, what we find is that in some of these fields where we've added these different patches, we actually have less grain left, more grain removed, than in fields where we didn't do that. Okay? And the arrangement seems to matter such that the many small patches have more seeds removed than the few large ones. So small patches of waste grain seem to be better managed by these mice than others. I'm going to not go through the conclusions here. And I've got just, I see I've run out of time already, but I'm just going to say one other thing. This is a corn seed in the mouse, in the mouth of or skull of Paramyssus maniculatus. I want you to look at that and think, what prairie seed is as big as that kernel of corn and hard as that kernel of corn? Dry field corn. That animal, that may not look like that mouth is open very far, but that's a really large gate. For that mouse to grab that, that's a large distance for it to spread its teeth. And then it has to apply lots of pressure to crush that grain or to cut through that grain with those very sharp teeth. Okay? This is a mouse evolved in prairies where seeds are small and they tend to be soft. Now they may be some hard ones, but they're still small. Okay? They've never seen one of these in their evolutionary past. And so you start thinking, well, what if these mice have changed over the years? What have they changed? And there's a technique out there which allows us now to go out and measure the shape of this skull in a very objective fashion. We can measure little landmarks where the teeth start and stop and the various points for muscle attachments and so on the lower jaw and the upper jaw. And we can do that with mice today. And then, through the through looking at museum collections, we can go back and say, well, what did the mice look like 100 years ago, a century ago? And we can compare their skulls and say, are these even the same mice that we had in the tallgrass prairie? And we can statistically do a bunch of magic with these numbers. We can create an upper jaw and a lower jaw like that, or the whole skull together. Those are all the points of interest. We can create measures then of what is the shape of this jaw and how does it change, and compare the skulls of mice from today to the skulls of mice from 100 years ago. And so if we think, what we have here are these dots representing those landmarks on the lower jaws of these mice in today's world, but on this frame which, if it was straight up and down and everything orthogonal, would be correct for the historical mice. Okay? So what you see here is when you see there's some stretching right here where this cell has been stretched this way, is to say that the distance between these 2 points has gotten longer over evolutionary time. So if we look at this surface, which should be flat, if the mice were unchanged over 100 years, these would be flat, nice square grids, everything looking normal. But in fact, where you see them stretched and squished, they've been changing shape. These mice are changing. They are not the same mice they were 100 years ago. And through the magic of statistics, we can summarize all of those points into sort of summary statistics, if you will, and say, well, are they doing the same thing in all these different places? We went to the Dakotas, we went to Minnesota, we went to Iowa, we went to Kansas, and we got samples 100 years ago and samples last year. And what we find is that in almost all cases, the evolutionary trajectory from what they were, being these white dots, to what they are, the black dots, is proceeding in the same direction. Now, what does that mean on this axis? Is it longer or smaller or whatever? It's all of those points. So I can't just say they've gotten, longer or shorter or whatever, but that they're all changing. And all of those are going the same direction, which is the key here, except for mice in Kansas. Mice in Kansas, from the Kanza, if you're familiar with the Kanza Prairie, these are mice close to the Kanza Prairie, and a part of Kansas called the Flint Hills, where there's lots and lots of prairie still left, plus lots of agriculture and a lot of pasture ground. And so they've changed in a different direction. they're not evolving in the same direction as the mice that are living in this almost totally row crop dominated landscapes that we have here in Iowa and the rest of the Corn Belt region. They're going in a different direction. But otherwise, yes, they are evolving. They're changing very rapidly to become something that they never were. These aren't the same mice that we had before. So yeah, we've lost the mice that were in tall grass prairie mice. We now have something different. There's a lot of sort of implications of these things, and this sort of summarizes all of the ideas. But what I want you to think about is that these mice, if they have the capacity to change, this tremendous impact on the environment that we call agriculture. It's a gigantic thing that hit them like light speed, okay? Early 1900s, my grandfather in Jewell, Iowa, was setting tiles as a kid, draining wetlands, and I don't know if he plowed any virgin prairie, but he probably wasn't far ahead of the virgin plowing, if you will. To my generation, where that's been gone so far long ago that most of us don't even know what it ever looked like. We can only imagine it from description. But tremendous change, and yet these mice have been able to respond to it in an evolutionary fashion. I want you to think about the implications of that for something like climate change. We're really worried about climate change. I'm not as worried about climate change as I used to be because I realize these animals have potential to change that's far greater than I ever gave them credit for. If they can do this, maybe they can deal with climate change pretty well. So remember at least that these guys out there, the enemy of your enemies, if you will, the enemies of agriculture, might just be your friends. And with that, think a little bit about the fact that there's wildlife out in those cornfields, and they're doing pretty well. It's not a desolate, barren place where nothing lives. It's actually whole populations and communities of organisms that are getting along just fine, thank you. And with that, I'll answer a few questions. I'm sorry I ran over. Yes.
Did you track them in nearby pockets of prairie at all to see the difference in communities?
Track little pockets of prairie?
Yeah.
Not recently, but we kind of do that all the time. We used to be able to do it recreationally. Now that IACUCs exists, we can't. But we don't always tell them everything we do. So we're in and out of prairies, officially and unofficially, off and on for various things. But
So are the communities very different?
yeah, they're really different in terms of the relative abundances. So voles are very dominant in prairies, and deer mice are relatively not so dominant. You go to cornfields, it's the reverse. Okay, so we see shifts in who's important or who's abundant, but not in terms of the species that are there. Now, I don't think you'll find too many voles out in the middle of a bean field in the dead of winter. So when you do find those in the summer, they're probably just traveling through, or mice that are, or voles rather, that are doomed to an unhappy end, if you will. But still, there's lots of life out there, surprising numbers.
But what about the shapes?
What's that?
Like if you took the skulls from the prairie guys that exist today and compared it to the crop guys.
Oh, in terms of
Doing
the shapes?
analysis.
Well, you know, we've only done it with this one species, and we're just getting to the answers. I mean, John hasn't graduated yet. We're waiting on the final answer. In fact, I got those figures from him just this week. So we're just learning. But what was shocking to me was that they would change like that, all in one direction. What was also important was we didn't see a time sequence. We picked these places that were far apart, thinking that, you know, the things that are further to the east and maybe to the south would be the places that went agriculturally most intense earliest. And so we might see more evolution there than we would see in some of the western or northern places. And location didn't seem to matter. What really mattered was just simply time. Your old skulls are really different than new skulls, and old skulls from all over are more similar to each other than they are to new skulls from anywhere. So there's some directional selection going on that has something to do with agriculture. And we'll look into biomechanics of this and find out if in fact it is related to the musculature and so forth that would allow for a more powerful bite at a wider gape of the mouth. That's what we're expecting. But right now, those wire frame things, you can stare at them for a long time, and it doesn't make a lot of sense until you do the multivariate analysis. But we still need to work on interpreting it. But like I said, this is just a week old. Yeah.
So I don't study mammals, but I've seen mice eat things and they don't, those big incisors are not for chewing into corn. They usually get things in their side and they can kind of do that thing, right? You can do that when you're a kid. So I'm wondering, the incisors, I thought they were to break and so they could dig better and that kind of thing. So
No,
I mean, that just doesn't seem like the right, the way a mouse would eat
Yeah.
a piece of corn.
yeah, but
With its
in
front
fact, it is the way they start on these pieces of corn. Let me go back to one of these pictures here. This one will do, I think. So they can't get the corn in their mouth far enough to get it to their molars to start with. So they have to reduce it to chunks. Okay, and if you look at these,
Chew on the edge.
if you look at that, you see how that's been gnawed off? If you actually look at the stuff under a microscope, you'll see two chisel marks, okay, where they come together. And they chip off those chunks. And once they get them into chunks, then they can move them to the back of their mouth and grind them up. But molars are for grinding things that are small.
Did they hold it?
Hmm?
Did they hold it?
Yeah, they'll hold them. Yeah, but they can't hold them over
It's not
here.
gonna give me that, I'm not crazy.
Next time you got a mouse, you try to pull his cheek back so you can see his molars. You'll find that's pretty hard to do. But the other interesting thing to do is you notice, if you look at this, and when you go out in the field, this is universal. They always start at the embryo end. They take the embryo out, and a lot of times they don't even eat the big, bulky, starchy part of the seed. So presumably that embryo is more nutritious in some ways. I don't think it has more energy per gram, but it maybe has more
I think it might have more protein.
yeah, more vitamins and protein. So that's what they're going for, and they leave this big starchy thing behind. Maybe they eat it later. Maybe they eat it if they get hungry because they run out of other things. If the winter runs long and times get bad, but they don't eat it most of the time. But they're doing this, and if you watch them, they do it with their incisors. That's what they're doing. Just like a beaver bites down a tree. That's what they're doing with it.
One last question?
Yeah.
Two last questions.
Two last.
So you said from the older study that when the guy did the stomach dissections, you found they mostly arthropods.
Right.
And then you had the later experiment with like the choice between
Right.
foxtail or corn. Do you know like if it had a choice between arthropod or corn? Which?
No, because it's hard to put arthropods in a bowl and let them sit there, and if unless they're dead, and if they're dead, they might not taste as good. But I'd like to do that. But not all arthropods are going to be the same too. So late in the summer, there's these black crickets, ground crickets that come out, and they eat tons of weed seeds. And I suspect because when I tear a cricket apart, there's just like big globs of fat in there. I bet mice eat a lot of those too. But at that time of the year, they maybe not quite as driven by that as they are by the cacheability of the organism, and crickets aren't very cacheable. So I don't really know what they're doing with the insect. That said, Tatiana's got a data set that we haven't even begun to dive into yet, but she's been trapping mice year-round, and 20 mice every 2 weeks, all year round for the last year, and it'll be 2 years this December, and she's pulling the stomachs out of them. And we're going to find out not what they prefer, but what they actually eat. So we might find that the diet is that they're full of insects at some certain time. Doesn't necessarily mean they prefer them, but that's what's available. And what we'd really like to know is if we see what Bill Clark seemed to find, is that in the spring they eat lots of insects for some reason. We want to verify that that's actually the case by looking at this sample over the year and then see that it shifts to crop seeds or plant material, and then back to insects again. We want to see that cycle and see how that happens. And it might happen a couple of times during the year. There's also this kind of weird thing that's happened a couple of years where the middle of June, between June 10th and June 20th, that all of a sudden weed seed predation has dropped 3 years out of 4. We don't know why. We think it's just sort of some weird coincidence and noise in the data, but maybe there's some particular insect that emerges at that particular time, or I don't know, we can make up stories. We really don't know.
One last question?
I was just going to ask you about farmers
Yeah.
and ask you whether you've done any work with farmers or whether you've got any of them on board. I mean, are they going to believe you that mice aren't as bad as you said they think they are?
Yeah, so no, I haven't done much work with farmers. It is kind of fun though to go up to town. I live outside a little town called Story City, and I go up to the hardware store or the lumber yard, and in that picture, that little picture said "my enemy's enemy," whatever. was actually out of a trade publication that was put into John Deere's little magazine that goes out to all of their John Deere owners. And so suddenly I walk into town and it's like everybody knew who I was. And, oh, you're studying mice, and they knew all about me. I was like, how did you know this? I said, oh, you were in the John Deere magazine. And I was kind of stunned by that. So we have a little bit of dialogue, but not much. And that's why I was asking if there were farmers in the audience, because I'd really like to know a lot more about the farming. I'm particularly curious about what farmers feel about this problem of having Roundup Ready corn and soybeans as a weed the following year as opposed to a crop. You know, it's obviously great for some things, but at the same time, when it hits the ground as waste grain, then it's immediately transferred to the weed category. And I'm curious how they feel how effective they feel they are at dealing with it. Because when I drive around, I'll see lots of soybean fields in particular, which have almost no weeds in them, but corn sticking up all over. And so it's pretty clear that, at least in some cases, they're not very effective at that. I'd like to tell them that if we could raise, from the population models that Matt and some of his people have done, if we could raise weed seed predation about, it appears to be about 10% above what it is now, another 10%, from 80% mortality to 90% mortality, we would see weed populations plunge with no chemicals at all. And I would like to think, okay, suppose you as a farmer didn't have to buy Roundup, didn't have to buy sprayers, didn't have to spend your summer driving around spraying stuff, didn't have to pay for an airplane to spray. Well, those are doing mostly aphids probably. But anyway, wouldn't that make your life a lot better and more profitable? So I don't know how we can manipulate those mice to get that last 10%, but I know that 10% is the goal. That's the benchmark. If we get 10% more, we put Monsanto out of business.
More mice, definitely.
More mice.
Definitely.
More mice, or just hungrier mice, or both. But I'm not sure how to do it. I think, literally, if we could get the mice to feed on weed seeds earlier by having less waste grain on the ground, we might have a chance at this. But that's my best guess, and I don't know how to get less waste grain on the ground, because that's pure profit going away from the farmers. And boy, they don't seem to be terribly concerned about it. The amount of waste grain out there is phenomenal. I'll also say that the variation in waste grain out there is phenomenal. Some farmers are way neater than others. So if you wander around a few farms, you get access to a bunch of farms, you find tremendous variation.
Okay, thank you very much, Brent.